Hello, everybody. I am not Dominique, your session chair. She's coming next. My name is Dan Gorelick. I am the editor-in-chief of the journal Biology Open, which is published by the company Biologists. There should be a slide showing now of the editors of Biology Open, 10 of us. They're all academics, just like you. And we review and publish papers throughout the breadth of biology and biomedical sciences, from development, cell biology, physiology, plant biology, evolution. If you study it, we'll probably publish it. We're a sound science journal, unlike the other stable of company biologist journals. We're focused really on whether your conclusions are sound and are supported by the data, and we don't care as much about impact. Here are the 10 folks that have done an excellent job in the last year and a half when I've been editor-in-chief, working really hard. If you see them at the meeting, some of them are here, please stop and talk to them, introduce yourself. They'll be happy to talk to you about Biology Open, share their experiences. Speaking of shared experiences, how many of y'all, as authors, have gone through peer review? Why don't you raise your hands if you've gone through peer review? Yeah, raise them, keep them up. Okay, great. And now, how many of you think peer review is too slow? You can keep your hands up. Yeah. Okay, so us too, which is why we started an initiative called Fast and Fair Peer Review, where we are providing authors with a decision with reviews within 7 days of submission. Yeah, it's pretty cool. So we did a pilot study between July and December of last year, where every manuscript sent to 2 of our 10 academic editors went through the Fast and Fair review process, and all the other manuscripts that went to the 8 other editors went through the conventional process. And as you can see on the graph, we successfully reduced the average time to first decision with reviews to actually under 7 days. The median was closer to 5 days. Now, how do we do this? Our managing editor, Alejandra Clark, came up with this incredible workflow, tight deadlines. The editors stepped up and were able to make decisions quickly, and most importantly, we paid the reviewers. We pre-contracted them, and we paid them, and they were able to give us high-quality reviews within a short amount of time. Now, how do we ensure that they're high quality? Well, besides the editorial oversight, if you can hit the next button, please, you'll see that our rejection rate was the same. About 30% of the papers that went for peer review, whether conventional or Fast and Fair, were rejected. And so because that rate stayed the same, we're pretty confident that the reviews are of high quality. I welcome you to ask some authors that too. There are a few authors here who unknowingly submitted to Biology Open and were part of this experiment, and I think you can talk to them and see for yourselves. Next slide, please. If you'd like more information, feel free to go to the preprint where we have all this data available. It's currently under review. No, I'm just kidding. Not at Biology Open. But I will end by saying now we need your help. So we are expanding this initiative to all of our academic editors. And you can help us by submitting your manuscripts to us, right? If you are an author and you're tired of waiting, please submit to Biology Open. If you're a scientist and you want to make some money doing peer review, please come find us, and I'll tell you about the application process. We're, like the other company biologist journals, we're part of a community, and we're here to work with you to make peer review more transparent, fast, and fair. Thanks very much.
Okay, so hi everyone. I'm Dominique Bergman, and I'm one of the editors at Development, a sister journal. And I'm thrilled to be able to chair this session on conceptual frontiers. So we had frontiers in publishing, and now we're going to have frontiers across many scales and many different organisms. And I have the pleasure of introducing our first speaker. So this is Michel Milinkovitch from the University of Geneva, and how reptiles got their looks.
All right, so good afternoon. Indeed, I'm going to try to give you some satisfactory answers to that question, how reptiles got their looks. First of all, we are working mostly on the evo and physics of skin appendages and skin color in many new model vertebrate species, like snakes and lizards and crocodiles and hedgehogs and tenrecs and even elephants, that we are investigating the development of their skin and the biomechanics of their trunk. But let's go back to colors. So skin color in reptile is fascinating because it's much more diverse, for example, than in mammals, and also you have tons of different patterns like spots and stripes and labyrinths. And we were lucky enough quite a while ago already now to demonstrate how they make green color, which is of course very frequent in reptiles. And this is through a combination of absorption of some wavelength by pigments, like blue wavelengths being absorbed by yellow pigments, and then photonic crystals that are generated by these wonderful cells that are called iridophores that contain nanocrystals of guanine and that generate light interference, and therefore they function as selective mirrors and they reflect blue and green. We were also lucky enough to demonstrate that during the color change in chameleons, there is actually a photonic effect. They are changing the mean distance between these nanocrystals in this lattice of guanine crystals. And you can demonstrate this by isolating an iridophore and by making the cell swell or shrink by using osmotic pressure, and you can give the cell any color you want. And for the species that don't change color, you can actually mechanically push on the skin and you can compress the tissue and therefore the lattice of nanocrystals, and you get a blue shift indeed. So all this is not possible without, of course, structural colors. All right, but we are mostly interested into patterns, and we investigate this wonderful species, which is a corn snake. That's the wild type, but we have access to monolocus mutants, many of them that are generating different colors or different patterns. This is quite cool because these variations recapitulate what you observe in terms of variation among species. This is a work which is fully directed by my wife, Anja, who is co-directing the lab with me, and she has been establishing families of 16 morphs over the years, generated a lot of genomic data, single-cell sequencing, and she's identifying each of these mutations and mapping it very efficiently. Just so that you appreciate the diversity of the snakes that we are breeding, in the lab, we have, for example, these 16 morphs with very different patterns and different colors. Tanazia also set up an amazing protocol for CRISPR-Cas9, and she generated the world's first gene-edited snakes, and now she's doing it routinely. This study was about the development of scales, but she's also using this tool to investigate the formation of different color morphs and different patterns, as you can observe. All right, so now if we speak about patterns, obviously you think about Turing, right? And indeed, since the wonderful work of Yanni in Tübingen and of course also Shigeru Kondo in Osaka, we know today, and of course the work of many other people, but these two people were really pioneers. They really demonstrated that the pattern of zebrafish, for example, is established through these cell-cell interactions between iridophores, xanthophores, and melanophores, and then Shigeru has been convincingly showing that the system is behaving really like a Turing system, and it can be described with partial differential equations. So I guess most of you know the paper of Turing, which is an absolute masterpiece paper, where he's demonstrating that even with two components, an activator and inhibitor that are diffusing in the medium, you can first of all, model this with two partial differential equations, and depending on the parameters of the system, you can indeed generate patterns. And in his 1952 paper, he's describing multiple dynamics, but of course the ones that are the most remarkable probably are those where you have morphogen concentrations that are stable in time, but periodic in space, and indeed with a single model you can generate, depending on the parameter values, you can generate spots, stripes, or anti-spots, for example. And today it is pretty clear that these Turing patterns are hugely relevant to biology, and they are explaining some of the patterns that we observe during morphogenesis. So, by the way, we have been publishing a review about reaction diffusion, and if you are, or should I say, if you find the mathematics of Turing extremely elegant but a little bit difficult to digest, I made a digest of the paper of Turing in the appendix 1 of that review. Okay, so reptiles generate indeed colors through the combination of pigmentary and structural colors, and reaction diffusion is an important way of describing the patterning. Now, in many species, the pattern is independent of skin scales. If you see, for example, this guy here, you see the pointer, yeah? In this ball python, it's like you took a brush, you put it in black paint, and you brushed, the animal. It looks like the pattern doesn't care about the position of the scales. But in many other species, the scales are monochromatic, like in this ocellated lizard, where in the adult, indeed, you have these beautiful chains of green and black scales. Now, the animal is not born like this, it's born like this. And this is exactly the same animal 4 years later. So, of course, it's not up to scale. So that's a difference of size. And what is important is that the pattern in the adult is definitely not random. If it was random, you would have some clumping. You can, of course, quantify this. But believe me, it's not random. And therefore, we wanted to understand what is the dynamic of pattern establishment, right? So we built a robotic system that allowed us to take pictures around the animal at very high resolution. You anesthetize the lizards, and you are reconstructing its geometry. And because you can do that at different time points across the life of the animal, you can every time using curvature thresholding, because our system generates a resolution of about 40 microns, we can identify each scale and its neighborhood. And because, fortunately, the number of scales doesn't change throughout the life of the animal, you can align the animal with itself at different time points. And then you can project this color of each of the scales for different time points, and then this is not a simulation, this is real data. You see indeed that the green and black scales are skipping, are changing colors. And the process is pretty long. It takes between 3 and 4 years. Okay, so now if the pattern is not random, that means, of course, that it's not scale autonomous. And what I proposed a while ago is that this is a cellular automaton. And a cellular automaton is an absolutely fantastic mathematical object that has been invented by John von Neumann in the 40s. And I don't have the time, unfortunately, to go into the technical details. And that's unfortunate because it's a mind-boggling work. But John von Neumann has been demonstrating that when you have a lattice of elements, in this case, it's a square lattice that he was working with. And we are working, of course, with hexagonal lattices. He was demonstrating that if each of these elements is iteratively changing state as a function of the state of its neighbors, then you can find rule tables that generate incredible complexity. And if this is true, given that we have the time series of color changes, we should be able to derive the rule, as indeed we could show that if you are green scale surrounded by no other green scales, then you have a 0 probability of changing color. But if you are green scale surrounded by many green scales, then you have a high probability of changing color. And the rule is very tight. This is data from multiple individuals and multiple time points. And you have a similar rule for the black scales. Now you can, of course, use this rule in a numerical simulation. So you get a cellular automaton simulation, and you see that you get a dynamic, you get dynamics, and you get steady state patterns, which are actually undistinguishable from real patterns in real lizards in terms of their bulk statistics, so neighborhood distribution. Okay, so this is cool because it means that there is a cellular automaton which is computing the pattern of the lizards on its back. But of course, all what we know about skin color patterning tells us that it should be reaction diffusions, and this is very different from cellular automata. So how comes? Well, the answer is actually pretty simple. So what I propose is that simply, the cells, they are interacting with each other in a thick layer of skin in the middle of the scale, but in between two scales, the skin is very thin. So you have to take into account this geometry. And what is happening if you are reducing the diffusion coefficient at the border of these scales? Well, this is something which is very easy to do. You take the PDEs that have been developed for zebrafish patterning, and you make them live on a domain where you take into account the presence of the scales. So you reduce the diffusion coefficient at the border of the scales. And when you do this, you see that indeed the scales are changing color homogeneously. So each of these hexagons could have different colors, but they are shifting colors homogeneously. And of course, this is very dynamic because when you change color, your neighbors can also change color, so you can again change color back because all your neighbors are becoming of the same color as you. So the system is very, very dynamic. Now, what we did more recently was to let the geometry do the job, because before we were using the trick of reducing the diffusion coefficient at the border of the scales, and how much should you reduce it? We were, of course, fine-tuning it such that it works. But now, we don't need to do this anymore, because if you use, for example, hexagonal prisms in volumetric data, you do get a cellular automaton behavior. You get a scale-by-scale coloration, and you can even use the real geometry of the real scales, and you indeed get a scale-by-scale coloration. Okay, so that's the first conclusion of the talk, is that this is actually the first evidence that the abstract concept of cellular automaton is not just this amazing and genius idea in the genius mind of John Newman, but is also a process that has been actually generated by biological evolution, and it emerges from the superposition of reaction diffusion with the geometry of the skin. Now, with Ibrahim Jahanbach, who is an absolutely fantastic computer scientist in my group, we have been showing that this process has evolved independently multiple times in different lineages of lizards and has generated dynamics that are very different in these different species and generating, therefore, very different patterns. Now, of course, the classical criticisms of this kind of approach is to say, come on, this is a very artificial model. You don't even know what are the molecules that are involved in the process. And the answer I'm usually giving, sorry to be blunt, but is I don't care, because the process is extremely robust to model variation. So what we're interested into here is the fact that you are discretizing a continuous system, right? And you get scale-by-scale coloration and cellular automaton behavior. You get that whatever is your reaction diffusion system, basically. You can use a Gray-Scott model, Schackenberg model. If you want, you will still get this discretization. And that's what is important. To push that even much more forward with Abul Zakani, who is a theoretical physicist in my group, he was investigating the fact that at the steady states, yes, you have green and black scales, but actually if you look at the components, the reaction diffusion components that characterize the black scales or the green scale, you see that you have subclusters. So you have different levels of greens, but these differences are very, very subtle. The point is not that you have different level of greens. The point is that the model suggests something extremely precise, extremely subtle. And you might say, yes, again, this is very, very artificial. Well, we did hyperspectral reflectance imaging in our lizards, and we see indeed that there are different levels of green, and that the level of green depends on the number of green neighbors that you have, exactly as the model is predicting. This is in Physical Review X a couple of years ago. And again, what is very important is that this subclustering is extremely robust. Again, you can use basically your favorite model. You're going to get this subclustering. So that's the, one of the most important take-home message is that biology, of course, is very messy, right? There is an unmanageable profusion of cellular and molecular variables. Despite this messiness, it can efficiently and quantitatively, that's what is important, be investigated mathematically, including with these very simple phenomenological models. And again, if you're interested into these kind of concepts, please have a look at our review. Okay, so now you can ask, of course, why is it so unreasonably effective to explain the color patterning in these animals? Well, it's very simple. It's because chromatophores interact. We know that as a fact. And we also know that the signals are diffusive. And there is no mechanics involved. And therefore, you only need reaction diffusion. Now, let me switch to skin appendages themselves, so hair, feathers, and scales. A while ago with Nicola Dipoy, who was a postdoc at the time in my lab and now is a group leader in Finland, we were lucky enough to put a very long controversy at rest. People thought that reptiles did not have an anatomical placoat and therefore scales in reptiles were not homologous to hair and feathers. Actually, they are. We did find the anatomical placoats in all reptiles. But now, in all these cases, in all these amniotes, there is one pathway which is absolutely essential for the development of the skin appendages, and it's the sonic hedgehog pathway. We have decided to play a little bit with this pathway using two drugs. One is SAG, which is activating the pathway, and sonidegib, which is inactivating the pathway. So we have been establishing a protocol, and many of the things I will show you now have heavily involved Rory Cooper, who is here, by the way, today, Ebrahim Jan Bach and Gabriel Santos Duran. We've been establishing a protocol where we are actually able to inject very precise amount of these drugs in the circulatory system of the chicken embryo. And it's allowed us to look at the effect of perturbing the sonic hedgehog pathway. So first of all, we injected SAG at E11, 11th day of development in the embryo, and we observed something absolutely amazing. We have each of the reticulate scales on the palm and digits of these chicks that are actually being transformed into feather buds. And you make just a single injection at E11, and then you get these feather buds. They develop into normal down feathers. And then when the animal is born with these down feathers, you wait long enough and you see that these feathers are going to cycle and generate bilateral symmetric feathers. So that's quite fascinating because a transient agonism of this pathway at E11 triggers a permanent transition, a permanent scale to feather transition in the chicken. And we have seen this Waddington landscape multiple times during this meeting, but that's for a good reason. It's a good analogy. And indeed, you have a path for developing reticulate scales. And by using SAG, you can kick in the system and push the development towards the development of feathers, and it stays there forever, and the feathers are cycling. You are not going back. Now, amazingly enough, if you are now injecting exactly the same drug, but 1 day later, so this was done at E11, and this is in Science Advances a couple of years ago, but if you do that 2 days, 1 or 2 days later, what you observe is something completely different. You see that this transient agonism 2 days later is causing a transition from a chemical pre-patterning of these little scales into a mechanical skin folding process. And this is in press in the other open biology, not biology open, but open biology. Now, what we also tried to do was to use sonidegib to inhibit the pathway. And of course, the idea was to say, what is happening now to the feathers if we're inhibiting the sonic hedgehog pathway? And what we observed is that we are shifting sonic hedgehog expressions from a spotty patterns to a stripe pattern. And we see that we fail to develop early on the follicular structure that characterize the feathers and their barb that are developing. And we temporarily produce proto-feathers-like structure that look like what some dinosaurs were exhibiting. This was published last week in PLOS Biology. And amazingly, actually, the system is recovering. So despite that the animals are born with naked skin in many regions of the body, you have actually follicles that are very deformed in the skin, and they recover, and then they finally produce feathers, and after 50 days, you have a nearly complete recovery of feather coverage. So in this case, the morphogenesis, the patterning and the morphogenesis recovers very robustly from this antagonism of the sonic hedgehog pathway signaling. So in other words, it's easy to transform the reticulate scales into feathers, but it's extremely difficult to prevent the feathers to develop, at least when you play just with the hedgehog pathway. So all this work done on the chicken, very heavily involved. Rory Cooper was an amazing postdoc in my lab. He left, I'm very sad. He left very recently, but that's good for him. He went back to the University of Sheffield, where he started as a new group leader in the School of Biological Sciences, where he will have two main activities. One is to work on the beautiful patterning and development of the scales in sharks, and in his free time, he will be the best fan of Sheffield United. Speak to him about it. All right, so, and I want to finish with an exception to this paradigm of self-organization through Turing systems for skin appendages. And the exceptions is, of course, the crocodile head scales. So more than 10 years ago, I proposed on the basis of statistical features of the pattern, if you're interested, we can discuss that afterwards, that the system is actually mechanical. And it took us 10 years to really understand what's going on. And again, Rory was heavily involved, Ibrahim, Gabriel, and also Grigory Timin So we published that just a couple of months ago, and what is happening is the following. First, we are using light sheet microscopy to image these very large embryos. As you can imagine, they are quite large embryos. And thanks Alain Chedotal for the development of these clearing protocols. They change our lives. So we are using a lot these protocols. So you see that the skin is smooth at the beginning of the development, and then you see grooves that appear in the scales, in the face of the animal. Sorry, I'm getting lost here. And you see grooves that are developing, they are propagating, and they are connecting with each other, such that you get, at the end of the development, you get what we call a scale, is a random piece of skin that happen to have a size and a shape that depends on how these grooves propagated and connected with each other. So you get elongated scales on the top of the head here, and you get more regular scales on the side and on the bottom of the jaws. All right, so now what we wanted to test was whether this is due to compressive folding. If you take, for example, two layers like the epidermis and the dermis that are attached to each other, and one of them is growing faster than the other, in this case the epidermis, for some parameters of the elastic material properties of these tissues, you can generate indeed polygonal structures. So to test this, we injected in the circulatory system of the crocodile embryo another drug, which is EGF, because of course it's well known to increase the growth of the epidermis and also its differentiation, so its keratinization. And when we do that, we observe that we get embryos that have much thinner and longer dorsal scales, and on the sides of the jaws you get this amazing labyrinthine pattern, folding pattern also on the bottom of the jaws. Very, very different from what you see in normal embryos. Now we make 3 injections at E55, 58, and 61, and what is happening if we let now, for 30 additional days, we let the crocodile develop normally, and they hatch, and what we observe is quite fascinating. Part of the pattern is recovering. So part of it is still labyrinthine, but you have anyway some polygonal scales that are forming. But these polygons are much smaller than in a normal Nile crocodile. And they are very similar to what you see in a Geitmann. So in other words, by modifying the mechanical properties and growth properties of the skin, you can transform a Nile crocodile pattern into a Geitmann pattern. Of course, if you continue injecting EGF, then at the end you get a newborn crocodile that is incredibly folded. All right, because we have all these data, we can segment it, of course, very efficiently. So we have the bones, we have the dermis and the epidermis, so we know what is the thickness of each of these tissues everywhere on the head of the animal. And then, thanks to Grigory, who has been establishing a new protocol for collagen staining, several of you have been showing images of second harmonic generation, which was indeed the state of the art for collagen. Is very, very difficult to image. We set up a very, very simple protocol that allows to get much higher resolutions. And if you're interested into collagen, maybe you can have a look at this protocol. And this allowed us, we use that for many things, but for the crocodiles, it allowed us to identify the orientation of the fibers everywhere on the head, and we see that they're oriented perpendicular to the main axis of the jaw on the top of the head, and you have 2 main orientations on the sides and everywhere else actually on the head of the crocodile. We also, of course, use EDU staining to identify proliferating cells, and we see that we have a variable growth map across the head of the animal, for example, very low on the top of the head. And we can then put all this into a mathematical model, the thickness of the epidermis, of the dermis, the presence of the bone, of course, the collagen fibers, and the growth map. Now, I'm not going to go into the technical details of the model, but it is a finite strain theory, a Neo-Euclidean material model for those who like these things. And we can feed it with the data that we get from our development data. Now, then you can do your computer simulations. On the bottom, you see the real embryos, and here in blue, you have your simulations. If you let the skin grow, you see indeed that you have grooves that are developing, they are propagating, they are connecting with each other, and they are generating these polygonal scales with long scales on the top of the head and more regular polygons on the sides and bottom, as in the real animal. Of course, you can play with these simulations. For example, you can remove the collagen fibers. We cannot do that in the real animal. And you see that you need these collagen fibers to get your elongated scales. And also, if you remove the collagen fibers, the scales are much smaller than in the real animal. And we can, of course, simulate the effect of EGF treatment by exacerbating the growth of the epidermis, and indeed we get this labyrinthine pattern that we see in our EGF-treated crocodiles. Now, maybe one of the most important take-home message from this is that by varying the elastic parameters like the shear modulus, the Young modulus, the bulk modulus, or shear modulus of the tissue, or the growth rate, you can generate any pattern that you find in the different crocodilian species, from Nile crocodiles to American alligators. So conclusion is that evolution has produced two ways of generating crocodile scales. On the body, it's like for hair and feathers, you have a Turing system, but on the head, you have a mechanical system that produces these head scales. Second, of course, our computer simulation and experiments show that it is actually compressive folding which is responsible for this mechanical self-organizational patterning. And we understand much more easily now how these patterns can evolve, and we can explain easily how different species have evolved different patterns. And of course, to me, what I think is one of the most important conclusions is that simple elastic models can recapitulate certain mechanical developmental process as reaction diffusion can recapitulate some chemical patterning processes. If you're interested in the crocodile story, you can go on my website. In our YouTube playlist, there is a movie that explains in detail the process in the crocodiles. Just to show you that it's not only specific to crocodiles, Will-Paul Dagenet, who was also a fantastic postdoc in the lab, she recently went to Curie in Paris. We have been showing in current biology last year that actually compressive folding is also responsible for the emergence of folds in the rhinarium, so the naked nose of dogs, cows, and ferrets, but with an interesting twist that we call mechanical positional information, so stiff vessels, stiff blood vessels under the skin are playing an important role in this patterning process. If you're interested, have a look at the article. So, of course, self-organization can occur through chemical patterning, mechanical patterning, boundary conditions, and now, like many other people, we're interested how these processes combine. So I thank a few reviewers, and most importantly, so Athanasia Tsika and we are really blessed with a team of wonderful people, theoretical physicists, developmental biologists, evolutionary biologists, mathematicians, animal technicians, lab technicians, a really, really fun group of people, and we are, I think, enjoying a lot what we are doing in Geneva. More information on my website, and thank you for your attention, and also thank for the invitation to share my work, and especially thank you, all of you, for these wonderful talks and wonderful posters that allow me to get less stupid by really learning so many things. Thank you very much.
Okay, so that was amazing. We've got time for a couple of questions, and also Michel has said that he will be around afterwards, so if we can't get all the questions, you can bug him later.
I also have a printout of the crocodile head with all the layers. It's pretty cool to see if you're interested.
Okay, so let's start with one.
Yes?
Hello, thank you very much for a wonderful talk and really amazing visuals. I was actually wondering about the concept of wounding and regeneration. So, for example, with the first model, you said that the number of scales is actually consistent throughout the lifespan of the animal and the pattern changes. If there was wounding and repair, how are the animals able to recreate the right number of scales and the right pattern of them?
So, unfortunately, they don't regenerate, so they scar. It's really a pity. We could test so many things by removing scale by scale and things like that, so it doesn't work, sorry. Yeah.
Okay. Thank you.
Hi, thank you for the amazing talk. And I found it very curious that when you apply the drug earlier, you would turn feathers into scales without changing the pattern. But when you apply it later, it would disrupt the patterning. So I'm wondering whether you can expound a bit more on how Sonic Hedgehog is involved in both making the pattern and starting the downstream pathways.
Okay, so you're starting me on something that is really, I find very, very important. Thank you for the question, but I will try to stay very, very short. So the problem, I think, is that we are looking at development and how genetics influence development too much into a single gene, view. Obviously, it's not a single gene that is involved. You have an extremely dynamical network of interactions that is changing through time during development. It just shows us that the network of interaction is very different at E11 and at E12, right? Because when you perturb it with the same drug, you get a completely different result. So not only the morphology is changing through time, but obviously the pattern of interactions. And if we really want to understand development, that's where we have to go. We have to understand the network, how it evolves through the development, also how it evolves in evolution, in between species. Otherwise, we will be in the fog for a long time. Yes.
Thank you very much.
Yeah.
That was fascinating, especially the cellular automaton model, the first example thereof. I was surprised that you have mitosis and proliferation happening at the same time as the cellular automaton model is taking place. So what happens to the sister cells that come out of a cell division? Because there's a proliferation that's happening as the cells are deciding, I presume.
Okay, I'm sorry, I don't understand your questions. We are looking at mesoscopic structures. So these are scales. They contain thousands of cells. We are looking at the color of the scale, okay?
So this is I'm talking about the border cells, which actually take the decision of the neighboring cells, right? You said the cells, the cellular automaton must
okay, so in the cellular automaton, it's called a cell. Von Neumann called that a cell, but he didn't mean a biological cell. This is just an element, okay, in a lattice. So we are looking at scales, skin scales, as being some of these cells, but they are not biological cells, of course, right? So now, if you're asking how these elements interact with each other, is that your question?
Yeah.
They interact through, at a much lower scales, now biological cell interactions, okay? These melanophores, xanthophores, and iridophores are interacting with each other. Short-range interactions is very easy to understand, right? The cells are bumping with each other, etc. Long-range interaction remains a mystery for a long time. How is that possible? Because actually, many people have been demonstrating that you don't seem to have diffusing molecules. But then, several people, and now I forgot the name of the main lab, I'm sorry, it will come back. But they demonstrated that actually you have a middleman cell, which is wonderful. It's like a macrophage-like cell, which is actually recognizing a xanthoblast, for example, little blebs, and then it is swallowing, it is phagocyting this bleb, but there is a tether which is maintained, and then the middleman cell is making a random walk, and there is this tether with this xanthoblast, right? And then when it encounters a melanocyte, it is spitting it out, literally, and you have a connection between two cells that can happen at long range. I'm talking about 5, 6, 7, 8 body length, right? So it is diffusive because of this cell, but there is no molecule which is diffusing, but the signal is diffusing.
Okay, thank you.
Okay? This is David Paricci who did that. Sorry, it's coming bad.
Okay.
Yeah.
So, and I'm sure other questions will be answered later, and you can also see the 3D printed.
Yes,
head.
of course. Thank you.
So let's thank Michel again. And our next speaker is Jonas Hartmann.
Let's get into it. So like many of you here, I'm very interested in the sort of cool things that cells, and especially groups of cells, can do when they work together. So be it in a sort of migratory group of cells that, let's say, migrates throughout an entire embryo, and while it migrates, it generates different organs, deposits these organs, patterns them, like in the posterior lateral line, in the posterior lateral line primordium of the zebrafish, which I worked on in my PhD, or be it the cranial neural crest cells, which also migrate throughout the embryo and also make cell decisions, etc. So when I say I want to understand that kind of thing, I kind of immediately get a bit stressed about it. because these systems are incredibly complex. Right? Already on the single cell level, they're incredibly complex, and then they come together and they work together. And so there is this complexity and diversity of these systems. So we have on the one hand dynamical complexity, so there's all these feed-forward, feedback loops, nonlinearity that create very non-trivial dynamics. On the other hand, we have many different molecules, so you have a kind of diversity of components. On the higher scale, many different cell types, and on an even higher scale, we have, of course, the sort of diversity across different species. So there's all of this stuff going on, so you know, how could we possibly understand that, right? So in my day job, so to speak, I work on sort of the idea of like maybe, you know, if we want to understand this complexity, we have to measure this complexity, right? So I collect lots of data, right? Like beautiful data, single cell data, doing like imaging the zebrafish, segmenting the cells. We get big data, segmenting lots of cells. We get, you know, multimodal data, integrating gene expression. We get like multi-scale, 4D, integrated, super whatever data, right? And this is cool, but the longer I've been doing this, the more I've sort of realized that really just because you measure everything in a complex system doesn't mean you can straightforwardly infer or understand how the complex system really works. So to sort of put it in a sort of nice quote, big data doesn't answer any questions or doesn't give good answers if you don't have good questions. So okay, but then the sun sets, right? So I get stressed about that in my day job, but then the sun sets and you can start to think a little bit differently, right? So what does it actually mean to understand the biological system? So we have a few candidates of what people are sort of doing, right? You can sort of do a genetic reductionism, you can do sort of a pathway reductionism, you can think about how things emerge from a dynamical system. You can just describe stuff in big networks. And I think we can all agree that the left two here are massively oversimplified to the point of usually probably being wrong, and the right one here is not really comprehensible for humans. I'm sure we can all agree on that, no problem, right? But even for the middle one, and in fact for all of these, the sort of one big problem that I have with that is that none of them really generalize. So all of them are kind of ad hoc explanations where I might explain something about the posterior lateral line primordium, I might explain something about the lateral, the neural crest, but like whether that sort of translates more widely or not is sort of, who knows, right? Maybe sometimes things are conserved, sometimes things are not conserved. I mean, you just have to kind of start over if you want to extrapolate. So for me, a really sort of central question then on the conceptual frontiers is whether we can at all hope to find human comprehensible and generalizable theories in cell and developmental biology, and whether we can develop conceptual frameworks to support those kind of theories. So that's a very big question, right? And I wouldn't be here if I didn't have at least some sort of answer to propose, okay? And we call this answer the core and periphery hypothesis, which we recently published, and this is work together with Elisa Gallo. And so, yeah, so what is this about, right? So to give you a basic intuition, there's essentially two ways of generating diversity. So one way is what one might call sort of the engineering paradigm, and we often think in this way, right? You take a bunch of Lego building blocks and you build them together in different ways to get different kinds of functions. So like sort of building a circuit. And you get kind of combinatorial diversity out of that. And the problem with that is that your building blocks might be general, so you can generalize over your building blocks, but the way they are assembled, the design principles, you have no expectation of them being general. But there's another way of generating diversity, and this is kind of the programming paradigm, where you take a system that is inherently capable of producing different kinds of behaviors, and you program it to do one of the many kind of behaviors that it can produce. So we call those, we sort of propose, right, that one might split a sort of a biological system into two parts. We call one the core, which is this highly versatile sort of part of the system that can have many different dynamics, generate many different patterns. And then on the other hand, we have a periphery, which is function specific and which essentially programs the core to do the particular function that is required. And when you have systems like this, of course, you can reuse the core again and again with different peripheries to produce different functions. So I'm not going to go into sort of more of the details here, but basically, of course, the idea that follows from this, right, is that if we have models and principles that explain how these cores attain their sort of generative potential, how they generate these many different different behaviors, then those kinds of models should generalize across the diverse use cases of the same core with different peripheries. So do we find that kind of architecture in nature? I mean, this is all good as sort of theory, and I think you just need to look to the talks that we have seen today to get an answer to that question. So, for example, at the molecular to cellular scale, actomycin is a great example of a sort of system that can do an enormous amount of sort of geometric mechanical computation and generate a huge range of different functions depending on the periphery, which might be its regulators, cross-linkers, but also the sort of geometry and external forces that impinge on an actomycin meshwork. So we've seen that sort of beautifully discussed today. If we move up to the cell scale but stay sort of with mechanics, there's this idea of differential interfacial tension, which basically sort of describes that when you have control over a cell's surface contractility as well as over a cell's adhesion to other cells, by just controlling these two factors, you can do a huge range of things in terms of moving cells around differently, creating different patterns, creating different morphogenetic events. So that's a sort of higher scale, but we see again this kind of idea of a central motif that allows to do many different things and is programmed to do many of these different things. And obviously, my job for this has already been done, so this is another beautiful example, the Turing patterning systems. But you can even take this sort of maybe, this is very sort of preliminary, but you can maybe even take this sort of to a whole embryo level and think about sort of broader strokes modules or like sort of like let's say a germ layer partitioning program that is sort of generally conserved, but then you sort of shape its periphery in order to create these different modes of gastrulation. We might hear a little something along these lines in a subsequent talk. So in short, we propose that versatile cores can be configured by specific peripheries to underpin diverse functions, diverse behaviors in biological systems. Theories of cores should generalize well across the different use cases of the cores and real biological systems. Not all of them, and this isn't the silver bullet that answers every question, but for a number of relevant biological systems, we can actually sort of make that kind of core and periphery decomposition. So I will very briefly go over a couple of things we can take from that, right? So perturbations, I mean, this is a very high-level kind of conceptual model, right? So we can't make very specific predictions, but we can say, you know, if you have a perturbation of the core, probably things will blow up. If you have a perturbation of the periphery, it gets difficult. You might change things. You might also blow things up if that sort of part of the programming is essential for the particular system you perturb. But one interesting prediction is that if you manage to take the core out and sort of make it work on its own, you should collapse to some sort of naive core behavior where it doesn't matter where you take that from. And so, in fact, like if we come back to our sort of actomycin example, that's exactly what happens, and that's what we've seen today. You see that in the wild, the core and periphery always come up together, right? You never see an isolated core in a real biological system, but instead they're together to do all these different things, but you take them out and they all collapse to this sort of dynamic meshwork, and you can put arbitrary or artificial peripheries back in to make it do interesting things. And this is another great example, right? So from the embryos, you maybe thought, okay, well, what's underlying the idea that gastrulation might be like this? But in fact, of course, with gastruloids, we know that if we remove cells from their sort of normal environment, but sort of maintain them just to still be alive, then no matter where you take them from, you get something very similar in a gastruloid. So I will skip also the considerations about evolution, right? But essentially, we have a sort of scheme by which we argue, by which we can argue that in evolution we can expect core and periphery systems to emerge, and once a core emerges, it would spread widely because it essentially enhances the evolvability of the organism or the population that has that core, right? So, and yeah, we managed to somehow publish this late last year. So if you want to know more, I would encourage you to go and read this. I've also written a bit about it on the Node. And of course, now the sort of remaining question is, how do we go from this kind of qualitative high-level hypothesis to like a more quantitative theory that we can really study? And I won't go into detail on anything on any of this, but one thing we're trying is to essentially come up with a quantitative way of formalizing this qualitative notion. and using what we call configuration function spaces. You can think of them a bit as like genotype-phenotype maps, and in this way you can actually formally define the compositions that match the sort of things we think are true for core and periphery systems. Another thing we're working on is to model the evolution of these systems using sort of simple evo-devo, like in silico evolution models of stripe patterning. And then there's many other things we would like to do if we find the time and resources to do them, bioinformatics search across data sets for these kinds of patterns, exploring links to ideas of computation, cell types, how this relates to modularity and the kind of Lego brick way of building diversity, and of course ultimately applying this back to the kinds of questions that I'm otherwise working on, right? Things like the swarm-like collective cell behavior of the neural crest, but also other questions. And of course I would encourage you to, want to read the paper and then to have a think whether some of your favorite questions might benefit from a reframing from a core and periphery perspective. And if you want to talk about this, get in touch. I'm always interested to chat. So yeah, and I should thank again especially Elisa Gallo, who has been a long-term collaborator in this, and also Roberto Mayor, who is my current boss, and who lets me do this obviously off the clock. And also to the BSDB and Company of Biologists for sponsoring me being here. Thank you.
So we can probably take one question.
I was wondering in the context, like how do you define where the core stops and when the periphery starts? Like for example, in actomyosin systems, you have like 200 actin binding proteins, and some of them you're going to need for any in vitro assay. So conceptually.
Yeah. So there's a long answer to this, but short answer, it's an empirical question, I think, and then it's possible that there may be sort of gradients, or that there may be sort of cases where you have layers where adding more components gives you more versatility, right? So that sort of expands, that sort of adds to the core, whereas when you add peripheral components, you reduce versatility, you're producing a particular outcome, right? So that's one way of defining it, and that can be continuous, but we suspect that in many cases it will turn out to have sharp transitions because of the sort of maturation that happens evolutionarily, where you stop wanting to make changes in the core because you're since it's involved in multiple processes, you will break things by optimizing it to do one thing or the other. So in that way, you sort of start to really localize mutations or control to some components and protect other components, right? But this could even go down to like to domains in a protein, right? So you could have domains that are core, that contribute to the core, but domains that are flexible or that are amenable to mutation and would be more localized in the periphery. There's a lot of empirical work that needs to be done there.
Thank you.
Okay, so thanks, Jonas. And our next speaker is Matthias Heinemann from the University of Groningen.
Thank you very much. I guess you all remember this book, right, from your very early university education. I wonder whether you also remember what the content of chapter 17 was. Yes, it was about this one. First half of the chapter, beautiful cell biology. Second half of the chapter, this damn complex regulation mechanism, CDK and everything. Well, my research is on metabolism, completely different topic. And when I read this chapter, I was always very disappointed because there was not a single mentioning about metabolism during the cell cycle. And of course, there might be good reasons for that, because how would you actually visualize metabolism, metabolites, metabolic activity? So I guess there's a good reason why we actually know very little about this. And this is something that we wanted to change, and we used budding yeast for that, and we had developed a microfluidic device through which we could follow cells through their whole lifespan under the microscope, individual cells. And then we have equipped our microscope with the possibility to also visualize a metabolite that is fluorescent by itself, and that's the reduced versions of NADPH and NADH. So what you now will see is here the trace of a single cell over a very long period of time, and then if you inspect this trajectory very carefully, you will see small oscillations here. And there's one oscillation, one per cell cycle, as indicated here by these orange lines, which is the moment when the bud, the new daughter, appears. Now, in fact, these metabolic oscillations really align with the cell cycle, and this is what we have shown by tagging V5. This is a cell cycle inhibitor with a GFP, And this protein would go into the cytoplasm at the cell cycle start. Then the moment when the bud appears would denote the onset of the S phase. And when this protein goes back into the nucleus, this would pinpoint to mitotic exit. So we now could use these cell cycle markers. Well, we see here a trace of a single cell as a function of time. You see this protein shuttling in and out of the nucleus. So we could see here it's in the nucleus, in the cytoplasm, then it shuttles into the nucleus, then it stays there for a while, then it goes back. And if you now superimpose this data with NADH here, which is these trajectories, what you can see is that this metabolic behavior is super nicely aligned with the cell cycle here. For instance, always at mitotic exit, this is where the trough in this NADH dynamic appears. So that suggests that at least in dividing cells, there's a strong coupling between metabolism and the cell cycle. Now, if I could look into your brain, you probably would say, yes, this is expected, right? There's probably different metabolic demands during the cell cycle, right? Just think about nucleotide biosynthesis during the S phase. But the surprising thing came when we plowed through this data. There's also cells that clearly show these metabolic oscillations, but there's no cell cycle whatsoever. This cell grew in size, but there's no cell cycle behavior. And that sort of suggested that maybe these metabolic oscillations are actually independent of cell cycle activity. And in order to test this, we had grown cells in the microfluidic device. We had seen their oscillations, that's a single cell here. And in this moment, we added a cell cycle inhibitor, stopping cell cycle activity. And I guess you can all convince yourself that these oscillations continue. And that led us to think that maybe there's something like a metabolic oscillator, a metabolic clock that goes autonomously from the cell cycle machinery. And it turns out that it seems that there really is such a metabolic clock and that this could actually exert cell cycle control. And the way we showed this is we had grown cells first on very low glucose, so they don't like it as much, their division time is very long, and at some point in time we switched to high glucose. So what you see here is the NADH signal in individual cells. So this is the population average, then there's some dynamic, and it reaches a different steady state. And now you might ask yourself, hey, where are the oscillations? Well, they're all hidden down here because we're superimposing many cells. If we now zoom into individual cells, they're shown here. Just each line represents an individual cell. And you could see when it would do its start, its early S phase, and mitotic exit. Start, early S phase, mitotic exit. And if you now focus on these cells here, you could probably all convince yourself that these cells would start at about the same time point. And their start is totally independent of when they have finished their previous cycle previously. And the indication that actually metabolism really exerts cell cycle control came from the following. You've taken these cells, NADH signal, averaged this, and subtracted the population level behavior up here. This is shown here. So the average of these cells is the blue line here, the black line, and this black line is the blue line here. We take the difference between those and get this. So this is now the nitty-gritty detail of what any of these cells would be doing. And what you now can see, if you look at the vertical green lines, this is when they go through cell cycle start. And when do they go through the cell cycle start? At the ascending phase of the NADH oscillation. And that's exactly what normally cells would do. So we think here that the start after the switch occurs at the ascending part of the NADH oscillations, irrespective of the previous mitotic axis, and so it's sort of waiting for metabolism to do the right thing. Now, if there is now this metabolic oscillator that potentially exerts cell cycle control, the question is now, of course, what would be the nature of such an oscillator? And it had been proposed that potentially respiration, the metabolic pathway of respiration, could have a role in here. So we asked, would these metabolic oscillations emerge from any specific metabolic pathways, such as respiration? And here the clear answer is no. And the no comes from the fact that whenever we have grown cells, and these are yeast cells which we can grow on many different carbon sources, whatever carbon source we've tested, we always found these oscillations suggesting that they would not emerge to any specific metabolic pathway. Now, where would they then else emerge from? And that shifted our attention to the question of, could they actually emerge from biosynthesis? Now, if you use your biochemistry knowledge from your first semester, then this you would recognize as the core metabolic pathways, glycolysis and TCA cycle. And of course, these pathways are not just there for energy provision, but they also would synthesize precursors that then would lead to amino acids, cell wall components, lipids, etc. And from here, the amino acids would get to proteins, from lipids we would get membranes, etc. So now, consider the scenario where these pathways here, these macromolecular biosynthesis pathways, would have a temporal pattern. And a pattern that would be segregated in time. So meaning where proteins would be made at a different moment than lipids would be made. That would mean that these pathways would pull precursors, including redox and energy cofactors, at different moments. Now, this could lead to the oscillation that we could see, but it came along with a challenge, and the challenge was that we had to measure synthesis rates of individual biomass components, such as proteins, dynamically during the cell cycle in individual cells. And that was not trivial. The way we started, we looked at first protein biosynthesis. We expressed GFP from a constitutive promoter and measured then as a function of the cell cycle the fluorescence intensity, which we used as a proxy for protein concentration, very carefully measured cell volume dynamics. And if we multiply here the fluorescence intensity by the volume, we would get the abundance of the protein as a function of the cell cycle, and then we would have to derivatize the signal, correct for maturation, etc. So a very noisy, very cumbersome process, but what we got to see is a diagram like this. And the way you have to read it, this would now be a cell cycle progression axis. You have here the protein production rate. Here, this would be G1 phase, and this is all the rest of the cell cycle. And the important message here, you can see two peaks of protein biosynthesis activity. Here, one at G1 and a second one. Now, as this is completely counter to what old literature tells you, and which was held against us during the review process, we had to come up with a second method, and I can't go into the details of this method, but it's totally orthogonal to this experiment here, and it gives exactly the same two-wave pattern. Now this was nice, so that meant that at least protein biosynthesis is dynamic over the cell cycle, and then we used the second method with which we determined this protein biosynthesis wave also for lipids and polysaccharides. And what you can see is that here, for instance, lipids biosynthesis has a totally activity pattern over the cell cycle. And so from this, we concluded that protein biosynthetic dynamics are temporally segregated, and we think this is what gives rise to the oscillations that we see during the cell cycle. Now, I already told you that it seemed that these metabolic oscillations would exert cell cycle control. And here I can in fact show you a concrete example of where we think how these biosynthetic dynamics exert cell cycle control. So what I'm now doing is I take this figure and just copy it so that you could see this would be now two cell cycles. And if you focus on this window here, this window would be the G1 phase. And what you would see now looking at proteins and lipids, now protein biosynthesis would ramp up, lipid biosynthesis goes down. And now if you hopefully think with me, what would that cause, what would that do with a protein such as an early cyclin CLIN3, which is, I could tell you, is a very unstable protein. So if cells would now ramp up protein biosynthesis, but there's essentially no more lipid synthesis, meaning the cell would not grow, what would that happen if protein biosynthesis ramps up fast enough? Then this would mean that this very unstable protein would really rise in its concentration. And indeed, this is what we see here. You see G1 progression, CLIN3 concentration really goes up as we progress in G1. And that leads, and this is what the last piece of data I want to show you, what you see, the color code here means on the single cell level, again, each trace is a single cell of this CLIN3 production rate. You can see that the rate ramps up, and then once it's at its highest point, then this is where the cell cycle would start. So, what have I shown you? Well, first of all, metabolism is not as boring as you might think. Metabolism is actually dynamic. Metabolism and biosynthesis is dynamic. I've shown you that this dynamic is independent of the cell cycle machinery, maybe hinting to a clock, and that this dynamic exerts cell cycle control. So if it's a clock, then our next big question, and I guess that keeps me busy until I retire, what makes this clock? So if there's this metabolic clock, what makes this dynamic in the first place if it's not the cell cycle machinery? And once we're done with this, then we probably have to go back to this chapter and add a bit of information on what metabolism would doing to the cell cycle control. Now with this, I would like to thank the 3 PhD students who had, whose work I had the pleasure and honor to present, Alex Papakinakis, Tonis Litsios, and Vakil Takaev, and the funders. And if there's anyone in the audience who would like to work with us on this journey, then I'm happy to talk to you. Thank you very much.
Excellent timing. So we do have time for a couple of questions. Maybe I can ask one first. And so you're doing this in yeast, and I'm a developmental biologist, so I'm very curious about whether, if you've got a number of different cells, whether you think metabolism ends up being coupled in those cells, or how do multicellular organisms deal with this kind of clock?
I don't know. The only thing that I know is that here, and of course you could even say budding yeast is an organism that asymmetrically divides, so I don't know how this would be, whether this is a phenomenon that only holds here, and an asymmetrically dividing organism. Maybe if I try to answer this question differently, for me, the CDK machinery has been always be so complex that I felt like this cannot just have fallen out of the blue and have accomplished what we now have as the cell growth and division cycle. So here the possibility would be that maybe with metabolism being probably in terms of evolution be quite old, that a metabolic clock could actually have done the same thing in ancient cells, which then has just been made more robust in modern cells with the CDK machine. But of course, I'm naive here.
Okay, how about the first one?
Hi, so I have two questions. In the SG2M phase, in the S phase, you're probably needing a lot more nucleic acid than G1 phase. So the metabolic cycle could be running differently because it needs nitrogen-based substances. If you arrest cells, that's my first question, it could simply be because of the need of the cell cycle, you're seeing a difference in the metabolism. The second is, have you seen in arrested cells, so let's say an S-phase arrested cells, is metabolism always peaking? Because that would mean the cell will just explode at some point.
What we see is we can dial down this metabolic rhythm by feeding cells with extremely low amounts of glucose. So what we first see is that these oscillations extend in terms of time. They get slower and slower and slower. And at some point, they're dampened. They're not there anymore. And then the cell cycle would stop. So it feels like metabolism, this metabolic clock, is sort of like an engine that goes and drives. And if it's strong enough, then it would drag the cell cycle along. For your first question, is at S phase. Of course, this is the phase where the nucleotides for DNA need to be synthesized. But DNA actually has a very, in terms of mass fraction on a cell, is actually very minute amount. So this is something that we would probably not see here. If I haven't answered your question to your full satisfaction, then you catch me in the break.
Yeah, really nice talk. I was wondering if there's, if you have looked at whether these oscillations are driven by circadian rhythm machineries, because a lot of these things, like protein synthesis, molecular crowding, they do generate rhythms independent of the cell cycle.
Yes, that was a question that I also got when we started with this 15 years ago. We have was a suggestion by a blind biologist. We have done experiments. I don't fully recall the detail, but at that time we have concluded that these oscillations would not be affected by anything that is circadian-like, and we mimicked it with light.
So just checking if there's a need to keep these two oscillations entrained, or do you see the metabolism just driving the cell cycle? Because if not, they need to be entrained, and is that effect on CLIN 3 or 1, sorry, I can't remember, is that sufficient to entrain it? It seems unlikely.
We have indication that, so CLIN 3 is an early cyclin, so meaning that the cell cycle start, and this I think is really the mechanistic coupling between a dynamic metabolism and the cell cycle. We have other indication of data through data that I haven't shown you, that there's seem to be also some molecular interaction towards mitosis, mitotic exit actually.
Yeah, because that's I think mitotic exit is so strong that it's amazing to think this could override that.
Yeah, yeah. You know, what I would love to do at some point, play with, use optogenetics and sort of dial down metabolism, make it faster, make it slower, and see how the cell cycle would follow. For this, we first need to know how metabolism is made dynamic in the first place. So that's why I probably mentioned that it would keep me busy until my retirement.
So our next speaker is Guillermo Serrano Nájera from University of Cambridge.
So tissue flows are one of the most fundamental physical processes that shape embryos. But they are very difficult to analyze because of their dynamic and complex nature. But fortunately, today we have very powerful analytical tools coming from the field of dynamical systems that allows us to compress complex flows into single organizing lines. So for example, this line here is an invisible barrier in the atmosphere computed from satellite data that clouds at either side cannot cross. So these kind of lines are called Lagrangian coherent structures, and they are used to solve many transport problems. For example, they have been used to predict butterfly invasion events across continents, and they have been used to improve our search and rescue operations at sea. So today I'm going to show you how we can bring these techniques to developmental biology and to study morphogenesis. And to do that, first I'm going to show you how to compute these material lines. Second, how they organize tissue flows. And third, I'm going to demonstrate how can they be used to in experimental design. But before diving in, this is a collaboration between two labs, the Matthias Serra's lab at the University of California in San Diego, who developed many of these analytical techniques and the mathematical models you're going to see, and Ben and Stevenson's lab at the University of Cambridge, where I am a research fellow. So we use avian gastulation as a model system that you have seen today already. For today, the chick embryo is just a flat layer divided into 3 territories. So we have in the, can I see? Yeah. So in the outside, we have the extraembryonic tissue that is expanding outwards, as we saw before. And then in the central tissue, this is the embryo proper. And at the back of the embryo, we have this mesoderm territory that has this crescent shape. So the cells in the mesoderm are undergoing 2 characteristic cell behavior. So on one hand, they are undergoing oriented cell intercalation, and on the other, they undergo individual cell ingression. And the combination of these 2 cell behaviors produce the convergent extension of the mesoderm territory into the midline, and in doing so, these cells push the tissues in the rest of the embryo, generating these large, beautiful tissue flows that characterize avian gastulation. So to study this process, what we do is we take time-lapse images, like these ones, and then we can compute the velocity fields using particle image velocimetry. And after that, what we do is we sit these virtual particles on top of the tissue flows, and from these virtual particles, we can compute over time what are for each of the neighborhoods of particles, how they converge or separate over time to find regions of maximum separation or maximum convergence. And these regions of maximum separation or maximum convergence are the Lagrangian coherent structures I said before. So how this looks like. So first, let's see the maximum separation. So we find that there are 2 reaches in this maximum in the separation map, and we call them repellers. So this line, repeller 1, separates the embryonic and extraembryonic territory, while repeller 2 bisects the mesoderm territory in two domains, an anterior and posterior domain. if we look for the regions of maximum convergence, then we have a single, what we call attractor line, that sits on the midline of the embryo. So essentially with this, you can see that we have compressed a very complex flow that changes over time into a single snapshot. But this is not the only advantage. These techniques are also very resilient to noise, and this allows us to compute all these properties in wide field images with embryos that have absolutely no label, they are wild type, and perform this kind of analysis in a high throughput manner. So now with these tools, we began to study what we decided to study first is what are the cellular mechanisms that give rise to these repeller lines, these lines of maximum separation. And so now in this project, now I'm going to show you how we can use these lines to rationalize experiments. So to address this problem, Alex Plum, a student of Matthias Serra, developed this mathematical model of the chick embryo where the embryo is represented as a compressible fluid where a nematic field that represents myosin produces the stresses that produces the flows, and also crucially includes the extraembryonic territory in the outside undergoing epiboly. Now if we compare the real embryo with the computational embryo, we find that these are remarkable similarities in the type repeller lines. So now as an experimental biologist, what I wanted to do is essentially knock out these lines. So my first intention was trying to understand how we could remove the repeller between embryonic and extraembryonic territory. So we noticed that the extraembryonic territory has very low active myosin in comparison with the embryo, and this makes us to think that maybe the extraembryonic territory is undergoing epiboly, but the embryo, because of this high myosin, is contracting, and this produces a tug of war between these two tissues that produce a line of maximum separation between them that generates repeller 1. So to test this hypothesis, what I did is I created a new technique to confine mechanically chick embryos in the plane, and in these conditions, epiboly cannot occur, so the extraembryonic territory is not expanding. And if we see the repellers, now repeller 1 completely disappears. And this is because the contractility of the embryo is no longer counteracted by the epibolic expansion, and therefore there is not a line of maximum separation between these two tissues. So now we have to tackle repeller 2, and if you remember, repeller 2 is a line of maximum separation that sits on the mesoen territory, and the mesoen territory is undergoing convergent extension during this process. So we hypothesized that repeller 2 essentially is a feature of convergent extension. And to test this hypothesis, that was kind of easy. I blocked the formation of the mesoderm by using a FGF inhibitor. And in these conditions, we get this very circular embryo. And if we see the repellers, repeller 1 is still there, but repeller 2 completely disappears. So far I have shown you that first we compressed these very complex flows into a single snapshot, and then we could design very particular experiments to knock out individually each repeller line. And we also can show that these flows have some modular properties, because if we combine this treatment and this treatment, then we block both repeller lines simultaneously. And not only that, our data suggests that repeller 1 essentially is a function of the isotropic myosin that sits all around the embryo. But repeller 2 is produced by a different population of myosin that has the shape of myosin cables spanning several cells that only sit in the mesoen territory. So what about the attractor? So the attractor is, we can receive the attractor. This is what I did during my PhD at the University of Dundee with Case Bayer. These are the attractors, as you can see. And if you want to know about this, please come to see me at poster 98. So I have shown you how I've been working in trying to reshape these embryos. And now I am working to try to do similar things in this time in vitro using gastruloids, by changing the boundary conditions or the periphery of the system, as Jonas said. And if you want to know more about this, then you have to visit poster 258 and look for Apolline, that she will be there to show you. So that's all from me. Thank you very much for the organizations to invite me and to let me to give this talk. And I'm looking forward for any discussion. Thank you.
So our next speaker is Anja Geitmann from the other side of the pond, from McGill, and hopefully we're going to hear some plants.
Thank you so much, Dominic. Standing between people and their drinks is a really unfavorable position, and so to make up for that, here's a horse. I'm going to talk about the link between microstructure and macroscopic trait, and the trait I want to talk about is the ability to move, and so I'm going to use that horse to illustrate what I mean. Of course, I don't need to tell you what makes a horse move. It's the muscles, and muscles are cells, and in the cells we have actin-myosin systems. So we have a multi-scale system of structures that ensure that the organism is able to move. Now, for those of you, and there's only a handful who are plant biologists, would know that I'm actually not working on animals. I work on plants, and I'm going to talk to you about moving plants, which is not necessarily a trait that people associate with plants, but I'm going to show you one that you know for sure, and that is this one. This is the Venus flytrap, which is able to move, fold its leaves in order to capture prey. It's actually a mechanism that allows it to feed itself. Now, this is a very fascinating movement. It's rather quick, and there's quite a few people who have tried to understand how that leaf is able to move. And these people who studied that came to the insight that similar to another system, which is happening in bladderworts, much smaller, you might not be familiar with it, it's actually a snap buckling system. So there's a pre-tension, and then the leaf folds in very rapidly in order to capture that prey. But there are other ways in which plants and plant organs can move, and you know probably some of those as well. So you've heard or seen of heliotropism, the ability of sunflowers to move their heads, as well as gravitropism, where a stem is able to bend into a different direction depending on the gravity vector. This video here illustrates a root actually growing in wiggly ways. That is a way of moving as well, and it's called circumnutation. Now, what these movements have in common is that they're based on a growth mechanism. And so to illustrate this very, very abstractly, what is happening, it's a stem or an organ that is elongating, and it's elongating asymmetrically along its length in alternating way, and that's how you get that wiggly movement, essentially. And so what's happening here is you get a differential growth or a differential stiffening that is happening. That is an expansion of cells through growth, but then in many cases also an increase in cell division on the side that bends, or that's on the outer side of the bend. Now, there's a third way of moving in plants or in primary organs, and that is independent of growth, which is when a stem essentially can wiggle back and forth without having to divide cells or having individual cells grow. And that mechanism is essentially hydraulic. It's based on differential swelling and differential pressure. And that system you might actually know as well. It's present in the mimosa, the sensitive plant. And this video here illustrates how quickly that plant and the leaflets of this plant are actually able to fold when you touch it. It's not accelerated, this video. And so a leaflet is able to fold when upon touch or another mechanical trigger or heat or cold within a couple of seconds, rather quickly. This movement is reversible, but it takes at least half an hour in order to get back to its original shape. So this is what I'm going to talk about for the rest of the presentation. And the question that we ask is, how are plants actually able to generate this kind of very rapid motion? And in order to explain that, I thought I'm going to give you, especially those of you who are not plant biologists, a quick primer on plants and plant tissues. So primary tissues in plants look more or less like this. They're very green in many cases, full of chlorophyll, but the important part in our context here now is that the cell walls which surround the individual cells are very, very thin. They're depicted here in green, barely visible, even though they're not green, of course. What's green is the chlorophyll. And primary tissues are under pressure, and that pressure is that of a bicycle or a car tire, or even higher. So turgor pressure in plant cells is extremely high. Typical tissues that would have these primary characteristics are leaf mesophyll or epidermal tissues or flower tissues. This distinguishes them from secondary tissues. I'm just add that because you might have heard of secondary cell walls, which is actually the materials that makes tree stems really rigid. These secondary cell walls are built on the inside. I'm not going to talk about that very much. Typically, these cells, when they're functional, such as in wood, are actually dead. So the functional cell is dead, but that is just on the side. So we're going to, for the rest of the talk, pretty much focus on these primary tissues and on the so-called thigmomonastic response of Mimosa pudica. Now, this motion is very dramatic. If you think of a lettuce leaf, and you bend the lettuce leaf 90 degrees, you break it, right? But this leaf here bends more than 90 degrees and does not break. So that's pretty cool for a plant. So that means there must be articulations in there. There must be hinges, and that is exactly the case. These hinges are called pulvini, and the mimosa leaf doesn't only have one, it has three hierarchies of hinges in there. And so you can see the primary, the secondary, and the tertiary pulvinus here, and all of those are able to move. The primary pulvinus is actually quite big, can be anywhere between 1 millimeter and several millimeters. The tertiary pulvinus, which you'd seen on that image before, is rather small, is at the base of individual leaflets. But together, these three hinges make it such that the leaf actually completely folds and droops down. What is the purpose of that? Of course, defense. It makes it unattractive to herbivores. So what does this look like on the inside? We see very conventional tissues here. In the center, we see the vasculature. Then we see the epidermis, of course. And then we have cortex tissue, which is a type of parenchymal tissue, so primary tissue, if you want. And what was known when we started this study is that somehow there is an exchange of fluid and pressure between one side of the pulvinus and the other side of the pulvinus, which then makes this organ droop down. So that was known. There's not many studies around that, but there was a study, a PhD thesis in the early 2000s that actually showed that the individual cells in the opposite sides of the cortex of the pulvinus shrink and increase in size. And so what we knew is there must be pressure exchanges, and the question really we pose is, how does turgor-driven swelling translate into this very directional movement into one direction? And so when we started this study, the postdoctoral fellow who was working on that wanted to inspire himself by a macroscopic example. It was the pandemic, so he was sitting in his kitchen and didn't have much else to do. Certainly didn't have a lab bench, but he had access to silicone tubes. And so he did some bricolage, handiwork, if you want. So he put two silicone tubes next to each other, put them under pressure, and then changed the pressure in both tubes, increased it on one side and decreased it on the other side. And not surprisingly, you will say the thing bent. All right, so essentially what's happening here, if you take one tube only, of course, and you put it under pressure, it will swell. If you connect two tubes to each other, you increase the pressure in one, it will swell. By not increasing the pressure in the other one or even reducing it, it prevents the swelling into the longitudinal direction and directs that swelling of the swelling tube into a bending movement. Now, interesting also now, if you pay attention, what we have here is, of course, an elongation in one direction, but we also have a whole lot of swelling into a direction that is not very productive. Right? Because turgor doesn't have a direction, it's a pressure, it's a scalar. And so swelling goes in all directions, not very efficient. What we asked then, or what he asked himself is, how could I optimize the bending, minimize the waste of fluid? Well, think about this for a moment, not very complex. You have to prevent the swelling. And so that's what he did. He essentially stabilized the tube in the circumferential direction by building in hoops and then measured how much pressure differential is necessary in order to obtain the same bending, or the other way around, how much energy is necessary in order to obtain the same bending. And he compared that, and indeed it was much more efficient to get a bending if he prevented that sideward swelling in his macroscopical model. So our hypothesis was, well, maybe the mimosa hinge, the mimosa pulvinus, has such a mechanism in order to prevent the sideward swelling, so to say. Where would it be? What structure would it be? We had a look. So let's go back to the actual structure of the pulvinus. And on the left side here, you can see now the scanning electron micrograph of the overall structure with the structures that I mentioned before. And what he did is he essentially did a few experiments. He wanted to look at the swelling behavior of this pulvinus. So he cut out parts of the pulvinus. He measured the length, the width, and the height of this cut out organ and then put it in different osmotic media to see what the swelling behavior. And what he was able to notice is that when he put it in hypertonic medium, there was indeed a swelling behavior, and it was very anisotropic, meaning it was preferential into one direction, and that was the long direction. Okay, so here you can see barely any swelling in height and width, but a strong swelling in the long direction. So that was pretty cool. Clearly there is an anisotropy to the swelling, and he hypothesized that it would be just in his silicon tube model the envelope that prevents the swelling in the other directions. So in order to prove that, what he did is cut off the epidermis. He essentially shaved it up and did the same experiment. And what he found is indeed that there's much less of an anisotropy in the swelling behavior. There's still some, but much less so. So that really coincides, or is at least consistent with a silicon model that tells us that circumferential stiffening would direct all the swelling into the long direction, which then causes the bending. Which structure might that be? Well, we always said it, it's the epidermis, but what in the epidermis makes it so? So we had a closer look at the epidermis, and here you can see the transition region between the regular petiole, which is the stele of a leaf, and the pulvinus region. And let's look at this in more detail. What you can see here is quite dramatic features, actually. If you look at this in more detail, you can, you can see that there are folds in the surface of this pulvinus. What does it make you think of? Folds on the surface of a bending thing. Well, for those of you who still lived in the era of plastic straws, this is what it is, right? So a plastic straw essentially prevents itself from kinking by having these folds and prevents itself from breaking while bending through these folds. So similar mechanism, essentially. There's a second phenomenon that was interesting, and that was at the cellular level, geometry of the cell. So if you look at the geometry of the cell here, very typical for a cylindrical organ, longitudinal cells. But when you go to the pulvinus here, you can actually see that the orientation of the cells is the other way around, perpendicular to the long direction of the organ. So that, again, is quite interesting. And I have yet another macroscopic model for that. Think of an air mattress, right? It's pretty easy to actually fold it up this way in the direction of the cells, where it's very difficult to fold it the other way. And so the mechanism, both at the cellular as well as the tissue level, definitely is favorable towards bending and making sure that there is no breakage while the bending is happening. Okay, so epidermis is the first answer, and that's really great. And so his idea was, yeah, it is probably the epidermis and probably the internal tissues, which look like bubbles of very, spherical cells, don't contribute anything to the anisotropy. So in order to prove that, he did a swelling experiment, osmotic swelling experiment, but this time only with the cortex tissue, only with these rather spherical cells. He cut out these cortex sections, put them in the same osmotic medium, and found, lo and behold, not at all what he was expecting. Rather than a complete isotropic behavior, he found still a strong anisotropic behavior of this very isotropically looking tissue. So that was puzzling. So we wanted to go about and understand what might drive these cells in the cortex tissue to swell in a particular direction, essentially. And so if you look at this from the inside, and here is a scanning electron micrograph where we take out the entire cell contents, the cytoplasm, we only look at the cell wall. And here's another mini primer on plant cell walls. They're not green. but they surround plant cells, and what you can see is that they are constituted mostly of polysaccharides, cellulose being among them. All right, and when we look at this with the field emission electron scanning electron microscope, we can see that these cellulose microfibrils show a dramatic parallel alignment, which is very weird. And here's another quick primer. Why is this weird? Because we don't expect it to be in these spherical cells. In spherical cells, we expect cellulose to be arbitrarily oriented. Only in longitudinal cells are they typically hoop-shaped. So here we had a spherical cell where the cellulose alignment was very strictly parallel, or rather perpendicular to the organ to the organ axis. There's a second feature that was remarkable, and those are these things, areas in the cell wall, which you would recognize if you were a plant anatomist. They're collections of plasmodesmata, essentially, or primary pit fields. Now, these primary pit fields, just to come back to this drawing here, are these areas where plant cells connect to each other. So they're plasmodesmata that connect the protoplasts. And not only are there holes, so it's perforated, but also the cell wall is thinner there. So that was remarkable. Well, not really, because there's a lot of water flow, but what was remarkable is the shape of these things. Rather than being round, which is what we typically see, they were oval-shaped or even spindle-shaped. So we did a little modeling of that. Imagine you have a material that is homogeneous except for these spindle-shaped, thinner, and probably softer areas. When you now pull on this material in order to test its anisotropy, what would you think? In which direction is it stiffer? Think about this for a moment. Horizontal or vertically? Well, we did some finite element modeling on that, and we found that the material is stiffer in the vertical direction. Okay, so through the presence of these areas of thinner cell wall, we also get an anisotropy to the material of the cell wall. So here we had a very microscopic phenomenon that contributed to the anisotropy. Now I have one final feature that showed us that there is more anisotropy to the feature, and that is the geometry of the cells. So when David, who did this study, observed the swelling behavior in the confocal microscope, he saw that the cells actually are not completely spherical, they're slightly compressed. And so when you then swell them, they go towards spherical, which gives them a direction. And since it was still the pandemic, this is how he explained it to himself, this is what's happening, right? Like an accordion sort of swelling up in a particular direction. And so all of this together now means we have three different scales at which things are happening. First, the epidermis, both the morphology, the tissue morphology, as well as the cell orientation. We have the cell geometry in the cortex that has an anisotropy, and at the truly microscopic, even nanoscopic scale, we have the orientation of cellulose microfibrils as well as these primary pit fields. So we have a nested and hierarchical system, all of which contributes to the anisotropy of the swelling behavior of the pulvinus, if you want. So cell microfibrils as well as tissue folds. Now, when I was writing this up, I was looking at this and I started asking the question, what about the actual size of the pulvinus? What about the length of the pulvinus? Does that make a difference at all? And of course, the minute that I asked that question, I realized that we got scooped on that very seriously by somebody who wrote this. And it is on the length and degree of curvature of the pulvinus that the amount of movement of the blade depends. Have a guess who wrote that? Well, Charles Darwin, of course. So sooner or later, whatever you do, you will get scooped by Charles Darwin, but nevertheless, we were able to publish this study on the mechanics of the bending of the pulvinus. We also have a parallel study where we actually looked at the signal perception of the mimosa, and together they give us this flow of information from perception to response. Now, to finish this off, you know that actually my main activities in my lab are not so much on mimosa in particular, or in bending or or turgor. They're on the cell wall and they're on the morphogenesis. So what is of course now interesting is how do we get to these different cell shapes, to their orientation, to the alignment of microfibrils, all questions that align with other research projects in my lab, for example, on the study of the epidermis of the leaf, or on a single cell system we work on, and that will be on the pollen tube. So with that, I'm going to conclude. I thank you for still hanging out with me here, despite the fact that the drinks are downstairs. I'm indeed at McGill University, which is in Canada, so if there are any Americans in the room who feel they should pay a drink to a Canadian, I make myself available. And thank you very much for your patience.
Okay, so we have time for a couple questions, and also after this we do have poster talks, so stay there for that. While people are finding the microphones, I'd actually like to ask a question, and it has to do a bit with the development of these. And so what's really striking to me is leaves grow from a particular direction, and the orientation, the cells are elongated in one direction, and you're going across the grain here. So it makes sense for the function, but how are they formed in that?
And that is yet another study which is underway now. We want to understand how you get to this completely orthogonal behavior of cell growth, which is not something that is very normal in plants. And I have a student who has a great idea that it might be related to organ boundaries, and actually it be an expanded organ boundary, but we're working on that and trying. It's actually quite difficult because the phenomenon develops very early on in the embryo, so to catch the moment where it happens is going to be a challenge, but looking forward to seeing that evolve literally, that study.
Hi there. So maybe I missed this, but it looked like the tissue was quite homogeneous all the way around. So do we understand how it just pumps liquid into the top, not the bottom?
And that's the other question. How does the same signal give two different opposing signals to the tugger pressure, right? Decreasing on one side, increasing on the other side. The short answer is we don't know. There is one single study that showed that there is definitely some water flow from one to the other side, but it's not very conclusive. It could also be from one side to the vascular system and from the vascular system to the other side. The other question is, how is that even possible in 2-3 seconds? Because plasma matter, while there are a lot of them, is that mass flow of liquid actually possible based on that? We know aquaporins might play a role in all of this as well, but surprisingly little is understood about this.
Hi, thanks for the talk. I would like to ask a question about evolution. So it seems like the plant is using several different measures to ensure that it bends in this certain way. And so do you think these traits are originating from like one single change during development, or do they arise like in sequence in time?
Very good question. So the mimosa is quite special in that it responds very quickly, is able to do that very quickly, and has this dramatic pulvinus. There are many other plant species that have pulvini, just not able to respond so quickly. They do that in a diurnal rhythm, for example, so it's hours going up and down, where the pulvinus structure also is less dramatic. So a great way to study that would be looking at different species to see which are the essential mechanisms for that movement. And so that is another study actually going on. Thank you for the question.
Thank you.
So I may have missed something, but the shape, the overall shape of the pulvinus seems to be that it's much shorter on the, I'm not sure what the right terminology is, on the axial side versus the abaxial side.
Yeah.
So would that actually predict that as you swell the whole thing, it bends? Is that sufficient to account for the bending of the leaf stem?
Yes.
I mean, I can't intuit it, so you might have to model
get
it.
what I understand what you're getting at. So differentials both would be swelling, but differentially because one is longer. Yes and no. What we do know is that there's a true reduction in, whereas there's an increase in cell size on one side, there's an actual reduction in cell size on the other one. So there is a deswelling going on. It's not just because it's less long, there's a true deswelling. So it's more fascinating than that.
And is the distribution of the little holes the same on that on one side versus the other?
We don't know.
Okay.
Yeah.
Thank you.
Okay, so let's thank Anja again. Okay, so the first flash talk is Megan Davey.
Hello, I'm Megan Davey. I'm at the Ralston Institute, which is part of the University of Edinburgh. My lab works on limb evolution and development, and I primarily use the chicken as my main model. So at Roslin, we collaboratively, between many groups, make transgenic and gene-edited chicken lines. I'm going to highlight one today, which is the chameleon transgenic chicken, which was published in Development last July. If anyone's interested in our new cell cycle chicken, which hasn't been published yet, I suggest going to see Zoe's poster, which is at poster 28. So effectively, you can either express Cre recombinase in the chameleon chicken. It's a Cre, it's a mini rainbow line, which is a Cre-inducible mini rainbow. If you either express or apply recombinase, Cre recombinase, then the cells will irreversibly change one of three colours, and you can follow the clones of those cells throughout development. So my lab has been interested in how the limb develops, in particular the forearm. The paradigm is that limbs develop from the shoulder towards the fingertips, but this is not supported by any of our current fate maps. And understanding how the forearm develops is really important because we have many different limb differences, which include a loss of radial or ulnar bones. So my student took the approach of applying topical Cre recombinase to our chicken embryos to try and fate map or improve the fate map of the developing chicken. In short, she discovered a lost membrane that no one had remembered about, the interosseous membrane, but we also discovered that the radius and the ulna come from completely separate areas. So we think now that the bones of the limb develop in a modular way and not in a progressive one. I'd just quickly like to use this opportunity to thank the company of Biologists for supporting the Egged workshops, which is a practical chicken embryology workshop that we hold. If you think the chickens could be good for you, you want to try any of these chicken lines, if you work in the UK, you can use any of the chicken lines that we've published, but you might want to sign up to the next Egged, which will be in 26 or 27. Thanks very much.
Next we have Jelle van der Mille.
Thank you. Yes, so I'm Jelle van der Mille at the Mitochondrial Biology Unit in Cambridge. And we previously found that neural stem cells in the fly larval brain, that they really require mitochondria for proliferation. And if you inhibit oxidative phosphorylation in these stem cells, they end up with smaller brains. And maybe that's not that surprising. But as a clinician, I also see patients who have mitochondrial disease. And none of my patients really have smaller brains. So it means that maybe the context determines whether a stem cell responds one way or another to mitochondrial dysfunction. And that's indeed what we see in the fly brain now as well. So neural stem cells only slow down their proliferation if many of them are affected by oxidative phosphorylation dysfunction across the brain. But if you only inhibit oxidative phosphorylation in a few neural stem cells all across the brain in a clonal way, they seem to cope really well and they proliferate almost perfectly fine. So it means that somehow the tissue must provide a buffer, a tissue-wide buffer against metabolic perturbation. And we asked ourselves, where could this buffer come from? And looked at the glia, which are shown here in green and form this large network across the brain that wrap around the neural stem cells. And Stavroula, who is a postdoc in the lab, then conducted an ambitious genetic screen to look for genes in the glia that could provide a buffer against non-autonomous OXPHOS dysfunction. And I won't go too much in any of the details, but one of the things we found is that lactate dehydrogenase, which is a glycolytic enzyme, is a glial gene that is required to buffer OXPHOS dysfunction in neural stem cells. And the model we have now is that these glia use lactate dehydrogenase to somehow crowdsource NAD from all across the brain and bypass mitochondrial oxidative phosphorylation in the neural stem cells nearby. And I mainly would like to thank all the people in the lab, and would just like to point out that we have quite a few positions open, mainly clinical positions, but do scan the QR code and come and talk to me if you're interested in mitochondrial dysfunction. Thank you.
So next up is Marta Marshalluk.
Hi everyone, my name is Marta Marchuluk and I'm a PhD student at Tristan Rodriguez Lab, and my research focuses on investigating the impact of mitochondrial DNA mutations on nutrient uptake and embryonic patterning. Why focus on mitochondrial DNA? Because it plays a crucial role in cellular energy production by encoding essential components of the oxidative phosphorylation system, OXPHOS. Moreover, mutations in mtDNA are major contributors to mitochondrial diseases, such as Delay syndrome, an inherited neurometabolic disorder, and mitochondrial respiratory chain disorders that lead to neurological, muscular, cardiac, and respiratory symptoms. Both of these conditions are in fact linked to the mitochondrial MTND6 mutations that we introduced into our human embryonic stem cells, which is a key component of complex 1 responsible for energy production in the form of ATP through OXPHOS. Interestingly, we observed that despite significant reduction in ATP production, as well as basal and maximal respiration, our mutant cells proliferated at a similar rate to control cells. Moreover, we found out that our dysfunctional cells were unable to self-organize and give rise to properly patterned tissues in 2D gastroloids. This indicates that the mitochondrial dysfunction induces metabolic reprogramming that compromises cells' ability to respond to signals that govern patterning. Thank you very much. If you'd like to know more, please find me in poster 96.
Okay, and then our last flash talk, Minita Desai.
Hi, I'm Minita from Swadhin Jana's lab at NCBS India, and I'm interested in understanding the process of cilia maintenance. As we know, the primary cilium is a specialized sensory organelle whose structure can be divided into the ciliary base derived from the centrosomes and the ciliary shaft. Defects in the cilia lead to a broad range of disorders called ciliopathies. And although majority of these are caused due to defects in ciliogenesis, which has been very well studied, a significant subset of these are late onset, indicating that defects in the maintenance of cilia can also be detrimental. Now, historically, cilia has been considered to be a highly static structure. However, studies in the past 2 decades have shown that the ciliary shaft is highly dynamic. However, there is nothing much known about the dynamicity of the ciliary base, so we wanted to address this gap, and we asked whether the centrosomal proteins that are so important in building the cilia might have a role in their maintenance. So we looked at various centrosomal proteins that have been well studied in the cycling cells of Drosophila and asked what happens to them after ciliogenesis in a differentiated cellular context. To give you a glimpse, we found that only a few, but not most, centrosomal proteins are present and maintained in the cilia. And if you remove them after ciliogenesis, it leads to resorption of ciliary structure and defective olfactory function, all of this without centriole loss, indicating that they play a crucial role in the maintenance of cilia. If you want to know more, please visit my poster, which is number 94. And with that, I want to thank everyone who has helped me in building this work. I want to thank the organizers for this opportunity, and thank you for your time, and I'll see you at poster 94.
Okay, so that's a wrap. So let's thank all of our speakers for another great session.